So I get the pleasure now of introducing our second plenary speaker, my friend and colleague, Amy Pruden, who essentially is one of the world's experts in the environmental dissemination of antibiotic resistance, and she's going to touch on that today. So, Amy.
So I'm going to talk about antimicrobial resistance. And I think there's a very strong case to be made that it is a societal grand challenge. And it definitely is very relevant to convergence. So we had very engaging talk yesterday about that, so I don't have to get too much into it, but for sure, we need deep integration across disciplines. Stakeholder engagement is going to be essential. There's a lot of team science involved, so I'm glad we're all here, science communication and ethics. So let's take a moment and think about, when is the last time you had to take antibiotics, or someone you love had to take antibiotics? What if it didn't work? Pretty scary thought. And that is becoming a more frequent phenomenon. And in fact, this article in the Lancet really did a thorough study of the infamous year 2019 and all of the deaths associated with antimicrobial resistance. And it's pretty grim and it's worsening. And so it is predicted that by the year 2050, deaths due to antimicrobial resistance will surpass those due to cancer. And so we have our work cut out for us. Let's talk a little bit about what is antimicrobial resistance. It's pretty challenging. So as challenging as SARS-CoV-2 has been to track in the environment, one little tiny virus, now we're talking about many different strains of bacteria. And to date, we have over 3,500 described antibiotic resistance genes, or ARGs, in databases. So we're not talking about one organism. So at the very root level, we're talking about antibiotics fail when we need them as a life-saving drug, right? From a microbiological standpoint, we're talking about that microbes grow in the presence of antibiotics. Some of them can even use them as a carbon source and subsist on those antibiotics. So that's what this Petri dish is showing. Those discs have antibiotics in it. If they're resistant, they grow right up next to them. On the right, we're looking at some of the molecular mechanisms. So they can pump the antibiotic out, they can degrade the antibiotic, they can change the target, for example, the cell wall. The point is, all of these mechanisms are encoded in antibiotic resistance genes. And the kicker is that microbes can share these genes through horizontal gene transfer. So our traditional engineering approach of killing or inactivating a pathogen might not quite be enough. We have to look at the DNA as well. So in terms of the grand challenge perspective, antimicrobial resistance is being framed as what's called a One Health challenge. So I just came from ID Week last week in Boston. It was all doctors, a lot of veterinarians. They're big into One Health, and it's a good thing because it's helping alleviate the finger pointing of, who's causing antimicrobial resistance. Oh, it's all the farmers using antibiotics, or it's the doctors that are overusing. Like, no, we all contribute and we all need to work together. So it's people, animals, environment. So the environment dimension has been getting more appreciation lately, and we're realizing that we have some catching up to do on the science. On the right is a figure that delves a little deeper into that environmental dimension. So you have reservoirs in humans and animals where antimicrobials are used and inducing selection pressure on the gut microbiota. But guess what? They're sprayed on crops too. And then if we think more broadly, not just antibiotics, but antimicrobials, fungicides, it's a big issue. They're being very widely used. And so these reservoirs in the environment can become enriched in resistant organisms and their antibiotic resistance genes, and evolution and dissemination is happening in all of these environments. And I've devoted pretty much the lion's share of my career to understanding antimicrobial resistance as an environmental contaminant. I got started back when I was at Colorado State University in 2006 and looking at rivers in Colorado that had a pristine origin, and you could kind of track the inputs of antibiotic resistance genes. So when UNEP in 2017 came out with this report identifying antimicrobial resistance as the #1 environmental challenge to be tackled, that was a big deal. And this figure in the report is a really nice illustration of how the water environment in particular is a key unifying thread. We have inputs from excretion of livestock and people and wastewater treatment plants. And we also have industry inputs from both pharmaceutical manufacturing, other industries. So it's really integrating all these sources. And then of course, water is life. I mean, it's a source of exposure for recreation, or if it's a drinking water source. And then most recently in 2023, UNEP just released this report that's more focused on, okay, what do we know and what do we do about it? And I was honored to be a contributor to this report. And here in the United States, we do have a national action plan for combating antimicrobial resistance. This is actually a requirement of all World Health Organization member states, and we're in our second iteration. And this second one especially starts to emphasize more that we need to be looking at these environmental dimensions of antibiotic resistance. So how do we look at antimicrobial resistance in the environment? So a challenge, like I said, it's not one pathogen or one gene, it's a plethora. And so that's where DNA sequencing technologies really hold a lot of promise, that you can do broad profiling. So a little bit about those, right? So we had a major game changer a little over 10 years ago when so-called next generation DNA sequencing came online. So what was first generation? First generation was cool too. That's Sanger sequencing. You could get about 2,000 base pair reads and very high accuracy, but you have to isolate the organisms first, and it's a one at a time endeavor. Whereas with next generation, you can take your air or water, soil sample, extract DNA, and sequence it. And so Illumina sequencing lets you get millions and millions of reads. It's highly parallel, but they're very short. You're at best going to get 150, maybe 200 base pairs. That's not a lot of information. And then this long read sequencing is being referred to as third generation. So that's your nanopore PacBio. It's really nice. You get long reads, so then you get more context. Is this ARG on a plasmid? Who is it in? Is it a pathogen? But it's a bit shallower. So there's always trade-offs with our methods. And so in terms of application, it's still useful to isolate organisms, the pathogen, because then you can get whole genome sequences and really compare at the strain level what you're dealing with and mobile genetic elements. A lot of what I'll present is this metagenomic sequencing. So that's what I was saying, directly extracting from the environmental sample, getting millions and millions of reads. Amplicon sequencing is where you first do a PCR reaction of the gene you're interested in. We saw some of this with the SARS-CoV-2 characterization, like the Arctic protocol, and you can zoom in on genes that you know identify the organism. And then there's a challenge, if we do want to look broader, we need to extract RNA and DNA. So viruses, right? A lot of the viruses we're worried about are RNA viruses. We did a monster literature review that I hope is useful to folks like you. We had an army of students doing a systematic review, just trying to see how these next generation DNA sequencing technologies are being applied to wastewater. And so we saw there's a lot of pathogen detection. functional gene profiling, antimicrobial resistance, bacterial toxicity, looking at toxins like cyanobacteria, and characterization of viruses. So a nice thing about metagenomic sequencing is you don't need primers, right? And so the viral world is largely unexplored because they don't have conserved parts of their genome that we can target with primers. So metagenomics in theory can really help with viral profiling. So again, it was a little over 10 years ago, next generation sequencing came online. And then a little over 5 years ago, Peter and I got a really nice NSF PIRE, which is the Partnership in International Research and Education. And we sent students abroad. So in this case, we went to Switzerland, Hong Kong, India, Philippines, Sweden, and the United States. And we did metagenomic sequencing of the sewage. And it's very interesting. And so we saw that if we ranked the numbers of resistance genes that we annotated in the sewage metagenomes, that they were quite low and depleted in Sweden, where we know they've been extremely proactive about policy to try to minimize the spread of antibiotic resistance. And at the other end of the spectrum in India, where maybe not so much. My husband's from India, I go there frequently, I can pick on them a little bit, but yeah, you don't need a prescription for antibiotics. You can just go to the pharmacy and pick them up. And this suggests to me that those kinds of policies and practices are reflected in the sewage. And so this is very exciting that we could monitor sewage for antibiotic resistance genes. And okay, we're thinking convergence here. So how can we connect this towards informing policy? And so as Peter and our students were writing this paper up, the lovely pandemic took fold and everybody became very interested in sewage monitoring. And so it's really an opportunity now. We've built it, right? We have the news, we have this infrastructure. And so in this paper, we delve into opportunities, how we can expand that wastewater-based monitoring to look at multiple pathogens, including antimicrobial resistance. And so just as we've been talking about, right? So we're talking about wastewater and the excrement and the antibiotics and antibiotic resistance genes and ending up at wastewater treatment plants. But broadening that a bit. We've talked about not everybody is connected to a wastewater treatment plant. Maybe we need to do some septic monitoring, and we're starting to do that as well. But how do we build this infrastructure to start collating the data, making sense of it in a way where those policy opportunities pop out? And so we do have another NSF project that's underway that we call Sewers. And this is led by our collaborator in computer science, Li-Ching Zhang. And so that's exactly what we're doing. So we're envisioning wastewater treatment plant of the future is going to have this cyber infrastructure. From the antimicrobial resistance standpoint, you could envision that doctors would have an app on their cell phone and they can see, oh, you know, these forms of antimicrobial resistance are really dominant right now in our community. I'm deciding between this antibiotic, that one. And that could be very useful information. And I did share that with the doctors at the ID week last week, and they didn't seem to think I was too crazy. So what we're doing now is trying to dig in a bit deeper, get some really high resolution data. So we've gone to our local wastewater treatment plant twice a week for over a year, sequenced over 100 gigabases. Oh, well, in this case, 37, later it's 100, but very deep sequencing. And also comparing wastewater treatment plants based on the demographics and the socioeconomics of the population served. So Virginia, we're here in Virginia now. It's a diverse state. This is lovely Northern Virginia, Southwest Virginia, little different. It gets pretty rural. We get some lower income communities and a lot of health disparities. And so the aim here is to start to get an idea of how that might be reflected in the sewage. And so one of our PhD students, Connor Brown, has been starting to dig into these data, and yeah, we've got a data problem. It's a lot of data. In this example, this is one way of looking at metagenomic data. We're annotating all of the antibiotic resistance genes. They're color coded by the class of antibiotics to which they encode resistance. And you can look for anomalies and blips in the radar. So we see a blip here that he was able to annotate and see that it looked like it was actually a cyanobacterium that was carrying certain antibiotic resistance genes that caused that increase. Now, that was annotating the metagenomic data for ARGs. You can annotate it for pathogens. And so here we're just comparing in Blacksburg and Christiansburg, Virginia. So Blacksburg is influenced heavily by college student population, whereas Christiansburg is the neighboring community. And so you can look and compare patterns in annotated pathogen groups. Now, here's where you get into a challenge with methods is metagenomics. And this is why we're doing deep sequencing. It's hard to get the resolution at the strain level. You can start to assemble those short reads, those 150 base pair, 200 base pair reads, and construct what's called a metagenome assembled genome. You can do that fairly confidently for dominant organisms, but it gets hard for rare organisms. Over here, we're showing how you can look at some of the other operational data like temperature and look at correlations. And so, yeah, we're focused a lot on trying to develop user-friendly tools to sift through the data and look at patterns. So one example is what we call meta compare. So it's a way of doing a comparative analysis of metagenomes and looking at the degree to which they're enriched, not only in antibiotic resistance genes, but antibiotic resistance genes on mobile genetic elements and within a pathogen host. And so we essentially get what we call a resistome risk score. And we try to be very careful about the term risk, because especially if I'm here with Carrie Hamilton, she'll school me. It's not technically risk in the sense we can't predict how many people might get an infection, but we can compare samples at a relative level and say, ooh, this one's got lots of mobile antibiotic resistance genes encoding resistance to really important antibiotics, and they're in pathogens. I think, you know, maybe I'll pay attention there. And so we're working on upgrading this pipeline. And actually, Monjura is a PhD student, and she's here at the conference taking a picture of this slide. She's got a poster, so I hope you can go visit her and talk to her about it. But basically, you know, since this is an index type analysis, getting a feel for the range, what is low, what is high, what do you encounter in various environments, and also kind of delineating between an ecological risk, like what is the risk that antibiotic resistance will evolve and spread in this environment versus acute human health risk, like, oh, there's pathogens that are known to cause disease in humans. So those are two slightly different questions. And one of the interesting things that she's noticed is that if you broadly look at these environments, so we've got like sediment, soil, wastewater treatment plant, human gut, that if you divide them into polluted, unpolluted, diseased, non-diseased, it is higher in the polluted, it is higher in the diseased category. So where we're going with this is we are trying to push the boundaries and see how metagenomics can be used as a comprehensive tool for monitoring multiple pathogens, antibiotic resistance genes in sewage environments. So we're fortunate to have gotten funding from the CDC to do this. And like I said, one of the big frontiers is how can you get strain level information from metagenomic data. So in this project, we are trying to make it very practical. And I think many people in this room have done similar experiments of trying to compare different sample collection, concentration, and DNA and RNA extraction methodologies. And so we've been looking at total nucleic acid extraction, which would be the ideal. You get your RNA and your DNA, and then kind of optimizing DNA and RNA separately. And then looking at, do we really need a 0.22 micron filter because viruses are smaller and will trap more? Or if viruses and other small particles are associated with large flocks, then maybe we can get a bigger filter size and get more water through there, get more sample, and still get those viruses. And so these are just some of the trends that we've seen so far. So here we're looking at the bovine coronavirus and MS2 on the top. And so interestingly, the green, the Zymo kit, which was total nucleic acid extraction, actually got us more viruses than when we specifically just did RNA extraction. So that's kind of nice because then we can do one extraction, have both DNA and RNA. And the Zymo kit also did very comparably with the fast DNA kit, which was just for DNA when we were looking at antibiotic resistance genes on the bottom. And then we've started metagenomic sequencing, and here's where we've done really, really deep sequencing to 130 base pairs per sampling, per sample. And so on the bottom, this is all antibiotic resistance genes that have been annotated, and then we're comparing the size of the filter and the kind of extraction kit. And basically we see that when we sample the aqueous sewage influent, which is what most people do, it does cluster nicely by the wastewater treatment plant. We looked at 2 wastewater treatment plants. So that's kind of what you want to see. But when we sampled from the primary clarifier, those primary solids, it clustered by kit instead of wastewater treatment plant. So then that means like you're clustering by your method, not your sample. That's not a great thing. We'll look into this more. It would be nice to be able to collect from the primary clarifier because it's concentrating the sample for you. So this is what we saw for antibiotic resistance genes. And then again, getting into trade-offs with methods, we're benchmarking the metagenomics with ddPCR. And so this is an example looking at the BLA-OXA gene, which encodes resistance to beta-lactams, is a major clinical concern. And what it's showing is that we do measure more genes actually with the metagenomics because it's picking up more variants. Because with ddPCR, we're picking specific primers, which are going to get a subset of those genes. And with metagenomics, you get the sequences of those variants. So that provides you with more information. But it's only going to provide you that information above a specific detection limit. So ddPCR has a really nice low detection limit. So we've been playing around to figure out what is the detection limit of metagenomics. And we've been spiking in internal synthetic standards. And so the good news is that metagenomics is pretty quantitative. So you'll meet people that say, oh, metagenomics, it's not quantitative. No, it is. But above a fairly high limit of quantification of 40,000 gene copies per milliliter. So below that, you'll get some sporadic detection here and there. It's just not quantitative. And then this is just to give you some appreciation for the complexity, if I haven't done that already. There's a lot you can do with these data. And so this is, again, Connor Brown, very talented PhD student in that he's transdisciplinary. You know, he gets the wastewater and engineering side, but he's also a biochemist and bioinformaticist. And so he's doing some real groundbreaking work assembling these data and building the networks and figuring out exactly which gene is carried by which mobile genetic or element and which organism. And so when you really zoom in on like this, you can see some interesting trends that you don't see when you just do broad profiling. So in this example, on the left, we have conventional municipal sewage. On the right is hospital sewage. And so we fed these reactors different ratios of hospital sewage. And it was only when he zoomed in like this that he was able to see that there are some specific pathogenic strains in the hospital sewage that enter the activated sludge that transfer genes horizontally, and you can actually track that. So next generation DNA sequencing. Yeah, so it's been an exciting decade. There is a lot to figure out. And so we've been trying to work with the community to try to establish, I don't know that we can say a standard method, but to work towards that, to look towards some standards, to at a minimum, report what you're doing to make the data as comparable as possible. And so Ben Davis and Ishi Keenum, who are now at EPA and Michigan Tech, they helped to lead this review to make those recommendations. And then the Water Research Foundation has been very supportive of this research and recently commissioned us to write a comprehensive report of the use of next generation sequencing in water and wastewater. And that's what funded that big monster review that I shared with you all. And they also funded a project specifically to look at monitoring of antimicrobial resistance markers in water and wastewater. And this was pretty exciting because it's basically the water utilities saying, okay, we hear you. We're sufficiently scared about antimicrobial resistance. What do we do? What do we monitor? And then, of course, as scientists, we're like, oh, well, we have to think about that. So we had an expert survey, an expert workshop, reviewed the literature, and basically came up with this framework. And it's a lot like what I've been hearing at this meeting that, well, there isn't one answer. It depends on your question. What are you trying to achieve? And so we identified 4 typical objectives that one might have. So the first is wastewater-based surveillance, essentially. So what is circulating in your community in terms of antibiotic resistance genes? But you also might want to know what's escaping your treatment process, and so what might become an exposure risk. You might want to quantify the removal achieved by your treatment process, so us environmental engineers can then do a better job designing those processes. And then the 4th goal might be to identify those hot spots where you think antibiotic resistance is at greatest risk of evolving and spreading. And then we have a lovely decision tree where you can pick what method depending on what your question is. And this gives you kind of a summary of what answers you might end up with depending on the question. A lot of interest in antimicrobial resistance on the global stage. So I'm pretty active with the World Health Organization and the Quadripartite Organization's initiative on what they're calling integrated antimicrobial resistance surveillance. And so the idea is very much a One Health approach. And we have some clinical monitoring and some level on farms, but a lot of catching up to do with environmental monitoring. So benefits would be providing that missing piece to One Health surveillance. Having this longitudinal data can then help us to identify what are the drivers of antimicrobial resistance, what are the hot spots of evolution, and that can help us to better focus our efforts and policy and mitigation and identify things like treatments that are going to provide the best barrier. And like I said, we could inform human medicine. Doctors could have an app. Veterinarians could have an app that guides them. And really, we urgently need these data to inform risk assessment and other models if we're going to move towards some sort of regulatory limits. Then there's the issue of data sharing. We're all going to have to grapple with this. You know, the data are only as valuable as they are available. but we're going to have to navigate the ethics of that. And so we need mechanisms that encourage sharing. So we've built the waterborne antibiotic resistance database as one forum where people can share their data. And the more we share, the better picture we're going to have of what the situation is. So expanding back out globally, in the UNIP report, what you'll see is they talk a lot about how antimicrobial resistance, like everything else, we can't address it in a vacuum. We need to think in terms of all the other world's problems. And for them, it's the Sustainable Development Goals. There's been a lot of thought and big brains into focusing on distilling what are major challenges for humanity. And so antimicrobial resistance is one that cuts across several of these, like obviously clean water and sanitation and access to healthcare and poverty. One of the quotes from the report is that AMR challenges cannot be understood or addressed separately from the triple planetary crisis of climate change, biodiversity loss, and pollution and waste, all of which are driven by unsustainable consumption and production. So we have our work cut out for us. And just to give one example, there are economic costs. So those of you who just care about money, well, you're in trouble too. And the World Bank provided a very detailed report explaining why that is. But essentially, GDP shortfall of 3.4 trillion US dollars annually, increased mortality, not just in humans, but in livestock, and that 24 million more people could fall into poverty by 2024. So what are we doing about it? A lot, but one of the most important things I think is training the next generation, right? So we need to think about our students, they're the future. So at Virginia Tech, we do have an NRT focused on antimicrobial resistance. It's called the SIPCAR NRT. You can read the acronym there, but convergence is the C in SIPCAR. So we're looking at integrating data science, environmental science and engineering, and policy, and stakeholder input from the very beginning, with the vision of informing policy and practice that's robust and likely to be effective for stemming the spread of antimicrobial resistance, and also to be adopted by stakeholders. And part of our program is the STEP certificate, so that's science, technology, and engineering and policy. So our students do earn this certificate. And so the idea is that they're immersed in this policy thinking from the very beginning of their training. And then, of course, all the things that we've been talking about are so important. So teamwork, science communication, but also, especially with the stakeholder engagement, that's part of what makes Convergence extra special, is really building skills, not just to talk, but to listen and to respect those that you're engaging with. And that's where we have to walk a fine line between science and activism. And we hold a lot of authority as a scientist, and we have to keep that in mind. And so I would argue that we need to strive to respect and love people with other viewpoints. Todd Schenk is our faculty member in policy who has been a real champion of this program in partnership with our NRT. And one of the methods that he's really a world leader in achieving this goal or working towards it is what's called joint fact finding. So you work together with stakeholders to agree upon what are the facts for moving forward. And my final plug is 2025, be there. I will be the chair of the next GRC focused on microbiology of the built environment, and it's going to be a lot of fun. So thank you very much for your attention, and I hope we can have some fun discussion.
Thank you for the excellent presentation. So I have a question on like the ARG or AMR in air. So I did see like a few studies on like the ARG in air, but not a lot. So I'm wondering if like the studying of ARG or AMR in air has like the significance in terms of health or like ecology. Thank you.
That is an excellent question and one very relevant to this group. So in our review, we identified some studies of ARGs in air, and there have been a couple papers recently that have been highlighting that, yes, if you sample the air and you do qPCR, ddPCR, or if you're able to get enough DNA from metagenomics, you will see ARGs. You also see some papers showing that if you're able to get a gradient of samples from like ones near wastewater treatment plants or ones away from urban areas, you see more intense signatures of ARGs. So I think though that it's a frontier area of study, and we need to try to identify the links, like is this really a source of transmission, or are the genes within organisms that have been killed by UV light. So I think Kim might have some data on that topic too. She kind of hinted during her plenary yesterday, so might be a good hallway conversation.
In terms of global questions of antimicrobial resistance, how do you see your findings moving to other countries, especially countries that won't have that much resources for metagenomics and all these super awesome groups and research?
Yes, that is an excellent question. And in the UNEP report, we were very, very mindful that we need to be global in our recommendations and not be leaving behind the low and middle income countries. So one of the issues is that people living in low and middle income countries tend to experience the problem more acutely. So water, sanitation, and hygiene is one of the biggest risk factors for the spread of antimicrobial resistance. So, people in these communities throughout the world might not have that degree of separation from their wastewater. And, so they're going to have higher infection rates. They're going to have greater need for antibiotics. And then ironically, there can then also be access issues. Like they can't get the antibiotics that they need, or they get the wrong antibiotics. And so it's a very tricky situation to navigate, that you don't want to be telling these countries stop using antibiotics, where they're like, hey, we can't get the basic antibiotics that we need help here. Like we have sick people that need treatment. And so there is sensitivity to that angle. Now, at the same time, we really need the participation of LMICs in this surveillance, right? Because we see that some of the organisms that evolve in ARGs, we can track them back to human travel. They picked them up in some part of the world, got on an airplane, and next thing you know, so BLA CTXM is an example of that. BLA NDM1, where you can trace it back to an individual that traveled somewhere, and there you go, genie's out of the bottle. So in these efforts to develop integrated surveillance of AMR, the strategy is to have a tiered approach. So have something that's low tech and accessible to everybody. So as you pointed out, metagenomics is not low tech or accessible to everybody. But what is E. coli, right? If there's one thing you can do, you can culture E. coli. And I've actually done it in the field. Like you can incubate it at body temperature, right? Like I've strapped little petri dishes to my body. I'm not saying that's a great standard method, but you know, if a lab has one capability, it's to culture E. coli. So the World Health Organization worked towards developing what's called the tricycle protocol. It's called tricycle because the three wheels of the tricycle are One Health. So that it was aimed at a protocol that's relevant to people, animals, environment, and accessible in low middle income countries. And so where they landed was E. coli. And okay, let's just take it a step further and add cefotaxime to the medium. So it's the same method, but now we just put a little cefotaxime in there and we can count presumptive, what are called ESBL E. coli, extended spectrum beta lactamase producing E. coli. And so I think that method is gaining ground. And then with some coordination, high income countries can pitch in and provide infrastructure like, well, if you can send us your isolates, then we can do whole genome sequencing. We can then start do comparison of strains and how those are mobilizing internationally. And then DNA extraction is kind of the next entryway. I think it's not that hard to extract DNA. You know, we have undergrads and high school students in my lab doing it every day. So if low middle income countries can extract DNA and then send it to some centralized resource where, again, high income countries are willing to pitch in, we can start working towards the metagenomic sequencing as well. Okay.
That was an excellent talk. I learned a lot. It was very inspiring. And you know what my question is probably going to be about, right? All the slides, like we're missing this one part of the environment, but I know you've written a review on it, the air. So are we just far behind on what we know about ARGs and air? Or, I mean, what are your thoughts on that?
So we are far behind. So why is that? I think part of it touches on your talk yesterday, that we've had a mind block to think that air is a pathway, right? We think we know the pathways by which microbes spread. And of course, water is more dominant than air, right? So I think we just didn't start to look there until more recently. And then we're seeing, oh, wow, there's a lot of antibiotic resistance genes here. I wonder what's going on. And we need a fuller picture. And then the other is the technical challenges that it is, I'm learning myself, not an air person, but working with really good air people. It's really hard to sample the air and get enough DNA to do metagenomic sequencing. But yeah, so we and others are starting to try to sort that out. I think Karen was first.
Thank you. Thank you for a really interesting talk. I'm really curious to hear more about your conversations with the clinicians in terms of if they were able to receive recommendations about what antibiotic resistance genes are circulating in their community. Do the clinicians see enough antibiotic resistance to be aware of the problem, and what were some of their interests and concerns?
So I can't say that my interactions with clinicians are super representative. It's not easy to break that disciplinary boundary. I think that's one of the hardest ones and most urgent ones that I hope with our NSF's pandemic preparedness, we can bridge that gap. Of course they see the problem. Like, so I've interacted with activist doctors at one extreme where they're like, we see patients dying. What do we do? And they picked up a paper in, environmental science and technology and say, oh my goodness, it's all coming from the wastewater. We need to do something now. Because I think in the medical community, used to patients showing up and you do something, right? You don't study it for 10 years. So that's at one end of the spectrum. But then the other issue, I think, is a lot of concern about liability. So one of the most important areas of research that's needed is looking at hospital wastewater. And there are a handful of studies. So for example, there's a study in the Netherlands and one in Germany, where you can track certain strains that were in hospital sewage, went through a wastewater treatment plant, and you can detect them in the river, right? Resistant to, and we've been doing some comparisons there, which is useful. And we've been developing some personal relationships with local hospitals, which is very, very helpful. But it's hard because their data is not necessarily in a format that's just ready for us to plug into our spreadsheets. So I think that's a real need right there, is to have better mechanisms for this kind of data sharing across the public health and clinical realm and the environmental realm.
Hi, Amy. Really nice presentation. So I'm just wondering, obviously you see the AMR accumulating in the environment, in the sewage and everything. I'm just wondering if this process is reversible, even.
Right, it's been hypothesized. If you remove the antibiotic, remove the antimicrobial, you remove the selection pressure, and now for the microbe, there's a cost of having a gene it doesn't need, and with time it will attenuate in the population. But what we're seeing is, you're right, it's not that simple. Sometimes that happens and sometimes it doesn't. It depends. And so I think what we're seeing, we have some examples now, like for example, in Europe, they were the first to ban use of antibiotics in livestock for non-therapeutic purposes, right? So they did that in 2006, and actually Sweden and the Nordic countries did it even earlier. So that's been a nice opportunity to kind of stand back and see, oh, what happened? But again, it's a bit complicated because there are some examples where you enacted that policy, but then antimicrobial use went up because the livestock were sick, and then that was used for therapeutic purposes, or there were loopholes. But I think a lot of that's been sorted out, like, okay, better hygiene, better practice on the farms. And now it really has gone down in most places where they've enacted those policies. And so we're approaching 20 years of that policy, and we're finally seeing some measurements that show, okay, resistance is going down. So in that example, it looks like it has taken time. So the question is, how long? And the other thing is too, again, these are life-saving drugs. So if we just say, oh, let's stop using them, there's really no point then, because that's the future we fear, where we can't use them anymore, right? So you have to, it's going to take something a bit more optimized. Like, how do we optimize the use in each situation so we minimize evolution and spread and keep the antibiotic working as long as possible? So yeah, we're hoping. There is a wastewater treatment plant that shut down in Virginia, and we got samples like before and after the shutdown. So that's going to be a nice opportunity to kind of see what happens, right? So if we really tell wastewater treatment plants, oh, stop discharging, no more ARGs to the environment. Okay, well, what happens? So there are opportunities like that.
This is CY Wu from Miami here. Yeah, so very interesting talk. I'm just curious, if I'm going to do air sampling, where would be the hotspot you would recommend? Because air sampling is not like a sewage. You have the flow, you get these spots you can do the collection. So for air sampling, we have to identify the hotspots to start.
Yeah, that's a great question. So Lindsay Marr and I actually have a project funded by NSF that we have samplers near wastewater treatment plants and livestock operations. So I think those are the usual suspects. And then the papers I've seen in the literature, similarly, like generally urban areas. There was a paper out of Sweden a few years back that generally looked at air quality index, and then a study in Hong Kong similarly, like looked at PM 2.5 and PM 10, which is quite interesting. So that's all to say, in general, these studies point to what we've seen in the water, that if there's more anthropogenic pressure, more pollution in general, you're going to see elevated levels of antimicrobial resistance. Along those lines, I think it fits with the previous question too. Another challenge is that it's not just antibiotics that induce selective pressure, it's antimicrobials. So that includes heavy metals and other compounds, and things that if we tell people not to use antibiotics, they might switch to something worse. And actually, when we do correlations with ARGs and antibiotics, antimicrobials, we actually usually see a stronger correlation with heavy metals than we even do with antibiotics. So again, we need a convergent, holistic approach to pollution in general, which will have lots of benefits. And one of them, I think, will be antimicrobial resistance.
So in wastewater surveillance, I feel like the question we get often is like public health action. What public health action will this data inform? And I'm curious if you think this data could be used to triage alternative therapies, something like phage therapy, right, which is specific to an organism or a strain or species. If you think that's a possibility for this data to inform that type of like resource allocation.
So are you envisioning, like, so if somebody engages in a phage therapy trial, so phages are an alternative to antibiotics, right? So they could go and kill the bacteria causing the infection, right? And then you didn't use antibiotics. In theory, they can't be resistant, right? So you're thinking like something more benchtop study or hospital level, or?
I guess what I'm thinking is like you have this for a patient, you have a very real and present risk in terms of they need a treatment. But we have this very distal hypothetical risk. And so clinicians, I think, are inclined to, well, I have this patient in front of me that needs treatment. And so I guess what I'm wondering is if we can inform the development of new tools, because there has to be something. And so obviously it probably takes a long time to develop these type of therapies. So
Yeah.
if we knew, like based on what we see in wastewater, who the players in this antibiotic resistance space were, if that might help us better, choose some candidates. I'm just trying to
No, I think that's a great idea. So, and I think we are kind of in that brainstorming stage of, okay, what at a concrete level, how can we better inform public health action? One of the challenges is innovation in general. So part of why we're having this discussion is we're not discovering new antibiotics. Like if pharmaceutical companies were just rolling out new antibiotics every day, then who cares? But they're not. There's some indication that we've discovered all the antibiotics that we're going to, for the most part. That's part of it, that we might be plateauing in what there is to discover. And then the other is the economics of it, that, for a pharmaceutical company, they're a business, like they can't just do this for free. And, people take an antibiotic and get better and that's it. Whereas, blood pressure and other meds, cholesterol, people take them long-term, they're more lucrative. So the bottom line is that you need some sort of investment. And the World Bank report and the UNEP report do talk about that and put a lot of the economics front and center. And it's not just investment in discovering new antibiotics, it's things like diagnostics. So a lot of times, we've talked about this, we don't diagnose what the person's ill with. let alone report it. You just try to get them better as soon as possible. But if we can more specifically say, oh, right away, this is E. coli and it's resistant to these antibiotics, this is the one to use, rather than sometimes the reliance is, ooh, we don't know what it is, so let's use a broad spectrum, hit it with a big broad hammer and kill everything. And then that's really a worst case scenario, because then you're putting selective pressure on the whole microbial community for no good reason. So diagnostics is an important area of investment as well.
Thanks for the talk. I was wondering, in your conversations with people in our community and your team and doctors and everyone that you've been talking to, if there's been conversations of what members of our community can do to contribute to trying to mitigate the risks, and thinking specifically about environmental engineers and those of us working in this, those of us in this room, in addition to surveillance, and if there's ideas that have come up thinking about control and mitigation.
Yeah, I mean, at the basic level, there's the CDC website, which we've learned is, not always the most accurate or effective information, but I think what they have on antimicrobial resistance is pretty good. Whether people trust it or not, that's an important dimension that we need to work on as scientists. And then, yeah, there's a lot to be said just for outreach type efforts. So one of the nice things about having an NSF NRT is you have an army of excited students. And so that's one of the things is they've been brainstorming some outreach type activities, and, we do things in our community, but I think sharing like what's successful, right? Like we have the internet, and so we can share modules and things that people can do within their communities. Yeah, because you're right, education's a big part of it, both the public and doctors, right?
I guess I was wondering also in terms of open research questions or things that still need to be investigated to try to, contribute to control strategies or, yeah.
Yeah. Well, I'm not the public health expert here. So, hopefully people working in that realm have a good idea of what's effective education and outreach. Yeah,
So I think we have time for just maybe one more question. Or two more questions.
So this is probably naive because I think your whole talk was focused on this, but how do we partition out or divide up and try to trace back the transmission pathways directly? Because we've been talking a lot about transmission in general, and it seems like there's still a knowledge gap there, if I'm understanding correctly.
No, that's an excellent question. And that's where I really think the field needs focus. Like we've got a mountain of evidence, like, yes, we know human activities and pollution in general elevates antimicrobial resistance measures in the environment, but show me the people who acquired a resistant infection because of their interaction with the environment. And so I think that's where the whole genome sequencing can be really helpful. And with all this lovely metagenomics I shared, I can say we're also putting a lot more effort into culturing organisms and sequencing their genomes and tracking them that way. And then there are some bioinformatic methods where we can look at the source tracking and try to figure out, okay, how did they get from point A to point B? What's the evidence of transmission routes? And then the other is, as Carrie talked about quantitative microbial risk assessment yesterday. And so looking at epidemiological models and trying to get her more data so she can figure this all out.
I was wondering how much we know about all of these individual genes in terms of do we know how they're integrated into other pathways? Are many of them just used for antimicrobial resistance, or they tend to be used for other things by the cell as well? And how much do we know about that? That
is a brilliant and insightful question. So you're absolutely right. It depends. So if you go to the Arctic tundra and do a core sample and pull up soil that was frozen before humans ever mass produced antibiotics, guess what? You'll find antibiotic resistance genes. Why is that? Well, most antibiotics are actually natural compounds. So microbes do produce them. They do have some exposure and they've evolved their own defense mechanisms. It's just that now we've mass produced them and like created a whole different scenario in the environment. So that is part of it. And then, yeah, so when we do analyses and we say, okay, we're going to compare to the comprehensive antibiotic resistance database of all the known ARGs and annotate them and put them in all those colorful categories I showed of which antibiotics they encode resistance to. That's a very 10,000 foot view of what's there. So that's where I was showing like, you know, you can zoom in on what you're interested in and get more specific information. So one thing is to take a next level cut and say, okay, which encode resistance to really clinically important antibiotics. So like we have sub databases of, okay, these are all the ESBL genes and the carbapenemase genes and, you know, things that encode resistance to antibiotics we know are really important to human health. And then sometimes you see a little bit different pattern, but a lot of times the overall trend is quite similar.
Thank you.
Amy, we just wanted to thank you again for this amazing and very insightful presentation. And thank you for sticking with us and answering all of these questions as well. Very grateful.
Thank you.